Hi everyone, thanks for inviting me along. And today I'm going to talk to you about our software, Servos 2. So this title is a little bit shorter than in the program book because it looked ridiculous when I put all the words on the title page, but it's basically software to accelerate annotation and segmentation of 3D image data. There's a little video just about to play. So if you don't know about Diamond Light Source, we're the UK's national synchrotron facility. This is a scan through a Australopithecus skull, which came from South Africa and is 3.6 million years old. And so this is sort of the flagship sort of tomography type work that we do. This was done on the I-12 beamline, which you may be familiar with. You may also be familiar with I-13. So for example, there's I-13C, which is Diamond Manchester joint engine beamline. And at Diamond, we also have Diad, which is where James works, which has two beams, so we can do imaging and diffraction at the same time, and do work such as the work that Fatih was talking about with the strains. We also have soft X-ray tomography at B24, so we use soft X-rays, and we can look at biological tissue in the water window between aqueous environments and carbon-rich environments. And as well as that, we also do electron tomography. So we have electron tomography on biological samples and on physical samples. So we have a wide variety of sources for our 3D data. So I don't probably need to explain what segmentation is to an audience like this. There's a description there if you want to read it. But just to know that it can be simple, but it can also be very complicated. So this is a GIF of some data that we collected on the Manchester imaging beamline of human placental tissue. So here you can see this work. Right. So we've got blood vessels. These blood vessels belong to the baby, the fetus. And around we've got some tissue. This is called villus tissue. And between these tissues, we have maternal blood volume. So basically these tissues are bathed in maternal blood. And this is where the nutrient transfer happens in the placenta. And I'll talk about this a bit more when we talk about the results of segmentation from the software. So this data isn't necessarily easy to segment. That's my point. Much. Right. So what is Servos? So Servos stands for supervision volume segmentation. This is an image of the first version of Servos, which some of you may be familiar with. And it utilizes unsupervised clustering of voxel data into supervoxels to simplify the problem. And alongside manual image feature extraction, so you have to extract various features from the image volumes yourself. We paint on annotations, and we use classical machine learning tools such as random forest and support vector machines to learn what label to give the other supervoxels. So I'll explain that in more detail. If we have our image of our bear and we want to separate the bear from the background, we don't necessarily need these thousands or millions of pixels there. You could also segment the bear from the background if you simplify this image into superpixels. In this example, you could say you started with 200 million pixels. You may reduce this by 3 orders of magnitude. And then, it's quicker. It's not only quicker to train a machine learning model on this, but also if you want to annotate on top of this, you have fewer elements to annotate. So annotating on super pixels which adhere to boundaries rather than pixels which don't. Then we extract image features using traditional computer vision techniques. So to try and enhance the difference between the areas that we want to separate. There are various ones you can choose from. We annotate the data via scribbles. So here I've scribbled on the nucleus of this cell and on the cytoplasm of this cell. Then we train a classifier. Basically, you group everything together into a matrix. N is the number of supervoxels, D is your number of features, and then we have our labels. So we may have 0 may be red, 1 may be blue, and -1 is we haven't given it a label. We use that to train our classifier. And then you predict on the supervoxels which you haven't annotated. And then afterwards you can do a sort of refinement. So for example, this red pixel here, maybe shouldn't be red because it's in a pool of blue, and you can smooth out those sort of artifacts. Right. So and at the end, you can export data. You can pick things, but you can pick labels, and you can look at, say, intensities and sizes and get some statistics out at the end to analyze your segmentation. So Servos was developed around 2013, I think, has started to be developed, and it's a standalone monolithic application, by Qt application. It's not straightforward to work on. I had to convert it from Python 2 to Python 3, for example, and it's not easy to collaborate on the same data sets. So we had a rethink. We wanted various things. We wanted switchable workspaces, deep learning techniques, because that's what all the cool kids use nowadays. And we wanted like ability to view multiple data sets on top of each other, like for correlative imaging. And we also wanted to work on some other stuff, so more bespoke stuff, like being able to run things in Jupyter notebooks using an API and so on. So we came with an architecture which was a client-server architecture, and we sort of, for viewing, we piggybacked off Nipari. So I don't know whether you've come across Nipari, but it's kind of, it's a Python multi-dimensional image viewer, and it's supported by Chang Zuckerberg money, so it's got quite a force behind it, and there are many plugins you can download and install. I guess it's kind of like a Python version of ImageJ. It's trying to be a bit like that. My colleague Avery did a lot of this work. She had a project where she needed to work on a particular electron tomography data set, pulling out virus particles infecting the cell. So the idea is, because it's a client-server architecture, you can run the server on a powerful machine, and you can have the client on your local normal laptop. And also, we have the ability to switch between workspaces and share data in workspaces. So this is kind of how it looks. I'll go through quickly because it's not that interesting to look at do, but you basically work through the tabs in sequential order, starting with extracting your features and your super regions, annotating, training a model, and so on. You have a sort of layer system, a bit like Adobe products, where you can put layers on top of each other to view data. You have data visualization tools, so you can switch between 2D and 3D views and so on. You have painting tools so that you can, paint buckets and paint brushes to annotate your data. Right. So I've talked about the standard machine learning models like random forest and support vector machines, which we trained in Servos 1. For deep learning segmentation, but these have problems. So you have to extract these feature volumes manually. You have to kind of know what you're doing. Each time you extract a feature volume, it has the same size as your original volume. You're creating a lot of data. There are generalization doesn't really work well, because you can easily overfit your model when you're training on a small number of features and you have relatively small training volumes. So this is a diagram you will have seen many times, and Rick's explained it much better than I can. But what you need to know basically is that this is a unit architecture, and you can divide it, say, into an encoding part and a decoding part. And this part is a feature extractor, basically, which then you can use to finally output a segmentation map when you put in an image. So we wanted to include something like this. We want to work on volumetric data. It's a 3D problem, but we actually chose to use 2D networks. So the reason we've done that is you can get pre-trained 2D networks. So networks which have been trained off, pictures of cats and whatever off the internet, you can use those. They're very good feature extractors already. And then it doesn't take much extra training, and you don't need much training data to get a good output. So the schematic on the left shows what we do. We basically slice the data in the orthogonal planes, create a data stack, augment it, and then train a segmentation model. And being generic here, it isn't necessarily a UNet anymore. And then when we predict, we take our data volume, which may be much larger, and then we do the same slicing. We predict volumes for each of the orientations. We rotate them back, and then we have to combine them. Which at the moment we do via maximum probability of but there may be better ways to do it. And this part I've separated out into a separate package, which is pip installable, which is used by Servos too. And this was published last week, but basically you can choose different architectures and you can choose different pre-trained encoders, and has a command line interface as well as the API if you want to use it. So here's an example of using Servos 2 to do some segmentation. So we combine these techniques, the supervoxel segmentation technique and the deep learning technique. So quickly running through, we usually denoise the data before creating supervoxels, otherwise the supervoxels pick up on the noise. Then we did some scribbles here. Kind of separate out the blood vessels from the rest of the data. We do our supervoxel-based segmentation, which is far from perfect, but it's roughly perfect. But then we train a unit on it, and we get a much better result. And then this is the original ROI, the region of interest of taken, which was 256 pixels cubed. And then we can predict on a much larger data set, and we get a reasonable result. This isn't perfect, but it's pretty good considering I did it quite quickly. And the idea is this saves you time, right? If you wanted to do this full size volume manually, it would take you a long time. And this took me an afternoon or less. So, and then you can view it in the viewer. You can do some rendering. Sorry about the color scheme, it's not the best. So going back to this example of placental tissue, we use units in Servos to segment the placental tissue, the same data, but we train two different models on the same data. So you can see my colleague Wynn annotated this. You have the blood vessel here, but you also have the tissue regions around the blood vessels. So we trained two models, one to separate out the blood vessels and one the tissue regions. And this shows the fetal tissue bathed in sort of maternal blood regions. So we have that. And then in combination with, when we combine these, we can take a look at the interaction between these two different elements in the data, right? So this is basically showing just showing the scales we could work at. This was an 8mm cubed tissue sample, and then we could zoom right in down to like the ends of the capillaries and the loops at the end of the capillaries, and you can see the sort of green sheaths is the villus tissue where the capillaries are. This is like a subvolume, and this shows the fetal villus tissue, and this shows the blood vessels within. And then we could do some modeling where you simulate applying a pressure at the top, and you could see how the maternal blood flows through here, and you have the flow lines here, and then you can see how they interact with potentially the flow rates and so on interact with the blood vessels within. So this lets you do lots of things. So yeah, that was just my one token example. But there are many more examples because it's supposed to be sort of agnostic to the imaging technique that you're using. So Servos has been completely rewritten. There are things I didn't talk about, like we have some import of data from like citizen science applications. So we had this, my colleague was working on a project where people clicked on all these virus particles in a volume, and then we had to import all that data and then use that as seeding points for other things. We can do various other things like clustering objects, and there are various other workflows, but there's still scope for adding more features, and we're open source. So if you want to add something yourself, then you can. If you want to tell us to add something, then we can talk about it. Finally, I'd just like to say thanks to all these people, and thanks for listening.